Thanks for your patience. So next, we've got a duet brought to you from the University of Queensland in Australia. And that is presented by Ashley York and Harriet Dempsey-Jones. The title of their talk is Identifying Cortical Columns for Slow and Rapid Vibrotactile Stimulation in Human S1 using 70 fMRI. Thank you.
Hi, everybody. Hi, I'm Harriet, and this is Ash. We're obviously presenting together, and we'll be talking about our work looking for cortical columns in human S1. So to start, what are cortical columns? Columns are cylindrical groups of cells that run from the surface downwards and contain neurons that are similar, have similar firing properties and similar connectivity. This is an example of 5 columns from rat vibrotactile cortex. Each one of these columns, the inputs represent one whisker. Columns have been identified now in so many areas across the cortex that some people wonder whether they're just an organizing principle of all of neocortex. However, there's actually been limited work looking in the somatosensory cortex to see if there is this kind of organization there. And that's something that we wanted to do, but there's been a little bit of animal work, which I'll tell you about now. So a group, Friedman and colleagues, they presented 3 different speeds of vibration to the finger pad of rodents. They presented 200 hertz, so very fast stimulation, with the aim of targeting specifically or predominantly Pacinian corpuscles in the periphery. They also presented 30 hertz stimulation, aiming to target rapidly adapting peripheral receptors, and 1 hertz stimulation, trying to target slowly adapting receptors. They used intrinsic optical imaging looking in area 1, although they've done a similar thing in area 3B. And they showed these patches of cortex that show a preference for the different kinds of vibration. This is 1 digit and another digit for the same animal. So this is somewhat suggestive of columnar organization. There's patches that show the same kind of representation. In our study, we wanted to see if the same organization could be found in human somatosensory cortex. So like Friedman, what we first wanted to do is look for patches that prefer different speeds of vibration. But second, we wanted to also examine whether these patches have depth down away from the surface, which is another important characteristic of columns. So we used 7 Tesla fMRI. We collected anatomical images using MP2 range sequence, 0.75 millimeter isotropic resolution. And for our functional data, we used a 3D EPI sequence and 0.8 resolution. And this is an example of our slab placement, which was centered over S1. So we collected data in 2 sessions. Our first session was an independent localizer task that allowed us to draw an ROI around the fingertip area. And to do that, we presented vibration stimulation to finger 1, then 2, then 3, then 4, then 5 in a phase-encoded design. Repeated that for about an hour. Then we submitted that data to delay analysis. This is an example for 1 subject here. We've thresholded all of those up to Q is 0.01, and then we manually drew an ROI around that. And within that ROI, we then looked for columns. So in our second session was our task specifically to probe for columns. We presented 3 hertz and 30 hertz stimulation in a block design, rapidly alternating between the 2 with about 2 seconds in between. And that session also lasted for about an hour. Okay, so from our anatomical images, we created a family of 11 cortical surface models. You can kind of see the different 11 layers there on the image. We discarded the outer 2 layers, the outer and inner layer, to remove some data that was most susceptible to biases from draining veins, and then the lower, which has the least amount of signal. And then we took our functional data that sits between layer 2 and 10 and averaged that in a node-wise manner. And this created our volumetric average, which we'll use for the maps that you'll see in the first part of the talk that I'll present. So we took this volumetric average data and we performed a GLM. I'm going to show you the outcome of the contrast for our 3 Hertz stimuli minus our 30 Hertz stimuli. Just to show you the color bar here, any nodes that you see that are red prefer the 3 Hertz stimuli, and any blue prefer the 30 Hertz stimuli. So this is what those look like. So you can see that there are some patches in that it loosely looks like what we saw in the animal work. What you'll obviously notice as well is that there is a predominance of 30 Hertz preferring nodes. Another thing we wanted to point out is that when we look at the beta values from the contrast, a lot of them are centered around 0. So while there is this preference, the preference is actually fairly low. Following on from that, we wanted to see which of these nodes showed a reliable preference. And to do that, we did a split half reliability test looking at our first 3 runs, our 3 odd runs and our 3 even runs. We computed a Jacquard index, which allows us to look at the spatial similarity between those 2 maps in terms of their preference. A 0 would indicate no similarity in the preferences of the maps at all, and 1 would indicate complete similarity. So these are the, these are the Jacquard values for our 10 participants. You can see they range from around 0.55 to 0.9. Importantly, we wanted to tell whether these values that we obtained are different to those that might be expected by chance. To do that, we did a permutation test where we scrambled the node preference and we created a null distribution. We did 1,000 permutations of that, creating a Jacquard index again. And this is the distributions of those values, and we can see that our values, our observed values, sit above those that we've observed there. So they're more reliable than we'd be expected by chance. For the rest of our analyses, we've masked out the unreliable nodes, and that's what these maps look like. Now on to Ash.
Alrighty, hey folks. I'm going to whiz through this in the interest of time, but I do encourage everybody, if you have any outstanding questions, bang them in the chat. All right, so at this point, we've identified some sort of patch or series of patches. They have minimal preference, but they do have some semblance of preference. So now we're going to move on to part 2, where we try and determine whether that preference extends across the entire cortical depth of our cortical ribbon. To do this, we've gone back to that family of 11 cortical surfaces, and for each surface, we've, instead of taking a volumetric average, we've taken an intersection of where the EPI values land on each of the nodes at each of the respective surfaces. Basically, we just went and made preference maps for every single one of those surfaces. And you can see on the left-hand side where you've got the volumetric average, that little guy, you've just got 1 beta per node. For the right-hand side, it's hard to catch it on the start of the surface of the cortex, but if you can see that there is much greater preference for 30 Hertz at the surface, where you start getting a little bit more representation of that 3 Hertz as you go a little bit deeper. All right, so we want to see how do these 2 hang together in a similar fashion. As before, we take the Jacquard index for every participant, pretty much just overlaying their volumetric average with each of their surface preference maps. And the best part of this is that you see the majority of the similarity falling in the middle portion of the cortex. This tells us 2 things. 1, volumetric average is not being particularly biased by any of the individual surfaces, which is fantastic. It's representative of that preference across the depth. And the other thing that's really cool is that the variability you see between the volumetric average and the individual depth map is centered around the bottom. The bottom here, where you have that weaker signal, much more noise, relatively greater noise, but you also see, without this mouse being particularly cooperative, you also see less similarity up at the very surface of the cortex, which again is exactly what we'd expect from a gradient echo sequence. So, determining depth profiles, do we actually see this preference running across the entirety of depth? If I take that volumetric average, I mask out those nodes that were not cooperative, they didn't respond consistently across odd and even runs. If I take the ones that do respond to the slow stimulation, and I keep them separate from the nodes that respond to the rapid stimulation, and then I look at the patterning across depth for each of those two groups, what I find is the most common pattern that we get for the slow preferring nodes is preference for slow across every single cortical depth. It represents about 20% of those nodes in that group. We see something very similar over for the rapid preferring nodes, where almost 50% of nodes are preferring that rapid stimulation from top to bottom of the cortex. Awesome. The more interesting part, again, the consistent variability that you're seeing, which accounts for about 15% for both groups, the consistent variability in that depth patterning is centered around the deep portion, again, where we're seeing weaker signal, but also up at the very surface of the cortex. Cool, excellent. We get some semblance of columnarity, but not complete columnarity. So, take it all together, we do find patches. They're a little weak, but they patches. Happy days. We do see a dominance of 30 hertz, which is something to be further investigated, I would suggest. But when we come back to the depth dependence of these patches, we do see some semblance of columnarity. Now, the most important part here, I think everybody needs to consider, what does the notion of preference actually mean within the context of identifying cortical columns? This is all very exciting. It's very enthusiastic, but again, we're in a paradigm where we force a preference. And thank you very much to our chair people who have been very liberal with my time. And we'll just also thank our co-authors, if the button will allow, and our funding body. Come see us at the poster.
Thank you very much. We will have time for one question. So, did you sample the same voxels multiple times across 10 layers?
Yeah, so the beauty of doing surface-based analysis is if you have, obviously, your voxels don't follow those surfaces. So the worst case scenario, if you're looking at a piece of cortex that's about 2 millimeters thick, and you've only got 0.8 millimeter sized voxels, you're getting potentially 3 voxels across that depth. For the depth-dependent selection of nodes, it was literally just a shoot through. It was whatever that data was that touched that node at that surface.
Thank you very much. There are more questions for you, so perhaps you can come up later, and let's thank the speakers again.